Hello everyone, this is Arya Suryavanshi from Department of Biotechnology, KLU. I am here to explain isolation of genomic DNA from plant cells by Ctab method. What is DNA isolation? DNA isolation is a process of purification of DNA from samples using a combination of physical as well as chemical approaches. The first isolation of DNA was performed in 1869 by Friedrich Mischer. Techniques utilized to separate DNA are dependent on the source, age, and the dimension of the sample. Generally, they aim to isolate DNA present at the center of the cell from various other cellular elements. Steps involved in DNA extraction. First, break down the cell wall and membranes. Second, centrifuge to separate the solids from the dissolved DNA. Third, precipitate the DNA using isopropanol. Fourth, centrifuge to separate the DNA from the dissolved salts and sugar. Fifth, wash the DNA pellet with ethanol and dry the pellet. Sixth, dissolve DNA. Principle. The Ctab method was invented by Murray and Thompson in 1980. It is a suitable method for elimination of macromolecules without affecting the purity of DNA. Plant cells can be lysed with ionic detergent Ctab, which forms an insoluble complex with nucleic acids in low salt environment. Under those conditions, polysaccharides, phenolic compounds, and other contaminants remaining supernatant can be washed away. The DNA complex is solubilized by raising the salt concentration and precipitate with ethanol or isopropanol. Material required. First, Ctab buffer. Second, TE buffer. Third, NaCl. Fourth, chloroform. Fifth, isoamyl alcohol. Sixth, centrifuge. Seventh, ethanol. 8. Electrophoresis apparatus. 9. Spectrophotometer. Preparation of Ctab buffer. To prepare 100 ml of Ctab buffer, add 1 gram of Ctab powder, 10 ml of 1 molar tris pH 8, 4 ml of 0.5 molar NaCl, and 40 ml of distilled water. Adjust to pH 5 with HCl and make up to 100 ml with distilled water. Procedure. First, collect fresh young green leaves and clean it with sterile distilled water. Second, weigh an amount of 25 mg and collect it into white. Third, grind the material with the help of tissue grinder in 400 ml Ctab buffer under cool conditions. Fourth, incubate the vial at 60 degrees Celsius for 30 minutes occasionally, mixing with gentle swirling. Fifth, centrifuge at 15,000 rpm for 5 minutes and collect the supernatant into a clean tube. Sixth, add equal volume of chloroform and isoamyl alcohol mix in ratio of 24:1 and 50 ml of ammonium acetate to 8. 7. Invert the tube for 5 times and centrifuge at 15,000 rpm for 5 minutes. 8. Collect the upper aqueous layer into a clean tube without disturbing the interface layer. 9. Reprecipitate the DNA by using double the quantity of chilled ethanol or isopropanol. 10. Centrifuge the content to precipitate the DNA. Discard the supernatant and air dry the pellet. 11. Add 50 ml of TE buffer and vertex to mix the content in the buffer. 12. Observe purified DNA on 1% agar gel after staining with ethidium bromide solution to ascertain its integrity. Conclusion. Orange color plant genomic DNA bands are observed under UV transilluminator. Applications. First, a determination of evolutionary relationship of a plant with its ancestors. Second, study of origin of certain plant species. Third, use in molecular biology. Fourth, used in recombinant DNA technology. Fifth, hybridization experiment of DNA. Thank you.